So we spent a couple of days thinking together about the nucleic acids and how these linear polymers of the nucleotides can carry information for either preserving it in the form of replication or copying it for transcription, ultimately for translation. And that is certainly the salient thing about the nucleic acids is their informational role. We're learning more about what RNA especially can do beyond just being information, but certainly the informational role of nucleic acids is absolutely central. The other major group of polymeric things, big molecules, that is crucial to life are the proteins. So the proteins are going to turn out to be an extremely versatile superfamily of molecules. They have many different kinds of functions, and we'll be seeing some of those as we go along at different steps. They are one of the classical examples of how structure reflects function. That is, the particular size and shape and charge of the molecule is going to make sense in the context of its function. And its function is not just what it does. Its function is what it does that is useful to the cell. There are things that molecules might do that aren't useful. Those aren't functions. Biology is the only science that is allowed to ask what is the function of something. We'll also be looking a little bit at how the sequence of the proteins leads to structures and therefore their functions. And in order to think about all of these things, we're going to use myoglobin and hemoglobin as models. They're not the only proteins we'll look at, but we're going to sort of have our first introduction to proteins through myoglobin and hemoglobin. They're both of them oxygen-binding proteins, and their structure will reflect that function of oxygen binding. And hemoglobin is capable of binding oxygen in a regulated way, and so it will be an introduction to how regulation can happen at the level of protein structure and protein folding. We have a couple of examples here of some proteins. This is a space-filling picture of myoglobin, which is not very it doesn't have any very obvious features when it's drawn this way, but it gives you a sense that it is actually a physical thing. It's not just an abstract platonic point. Here's a molecule of insulin drawn to a similar scale. I want to sort of give you a little bit of a feeling for some of the variety of proteins before we get into the how and the whether of it. There will be posted on the website a poster, and I don't have it in a projectable form right now, but I'll just hold it up and everybody watching the video can wonder. These wonderful color scale diagrams of a whole gallery of proteins. And they're a very good way to have an intuition for what protein shape and structure is like. So the ones we do have right here, we've got, for example, this is chymotrypsin, which is a fairly small protein with a fairly dumb function. It just cuts things up. Here's a somewhat larger protein, and we're going to see that that kind of pincher shape is related to its function. It's a DNA polymerase, and it's part of a larger molecule. Here we have maltoporine, and it's a transport molecule. You may have remembered that when we were looking at the genome earlier, how large a fraction turns out to be dedicated to transport, because membranes are impermeable without the selective transporters. Here's a picture of a fragment of collagen. If this were the full size of collagen, it would reach up to the ceiling. And we're going to be looking at collagen in a little more detail, but it already gives you a little bit of a sense of how this molecule might be involved in strengthening things. Here's plastocyanin, one of a family of things involved with photosynthesis. And photosynthesis is a very important set of reactions that we'll be talking about later on. Here's phosphoglycerate kinase, one of the glycolytic enzymes, and it too is going to be part of an absolutely central pathway of metabolism. So I think this should give you a sense that the proteins have actual shapes. They're not blobs. They're not sort of an indistinguishable mess of stuff. They have individual and particular shapes, and we're going to see how those shapes actually make a difference for function as we go along. The proteins are all of them made up out of amino acids, and protein structure only makes sense if you know amino acid structure. So I will expect you to know the structures of all 20 amino acids. It is not as bad as it sounds, and I'm going to walk you through them so you can see that knowing the structure of the 20 amino acids is in fact fairly straightforward, especially if you took me seriously at refreshing your functional groups. But the reason I want you to learn the structures of the amino acids is several-fold. First of all, it really is hard to talk about protein structure if you don't know what the building blocks are. Secondly, many of the amino acids are going to come up again in multiple metabolic pathways later on. So by learning these now, you're going to have a beginning of the vocabulary, the structural vocabulary you'll need to learn metabolism later on. So let's start looking at the amino acids and some of the ways of organizing them. Now, we've talked already about how an amino acid is something that has a primary amine group at one end and a carboxylic acid at the other end. That's the definition of it. There are 20 amino acids that are routinely incorporated into proteins. Almost all of them have some kind of a side chain hanging off of that middle or alpha carbon. Of those 20 amino acids, you can see that there are 8 that are clearly are clearly hydrophobic amino acids. All right, and if we look a little bit at the structure of these amino acids, you'll see why they're hydrophobic. And I'll also try to give you a taste of the coding for it. I'll probably use the 3-letter coding on a routine basis, but you should be at least aware that some places use the single-letter coding, and I'll try to make that a little more intelligible as we go along. So sort of the simplest hydrophobic amino acid you can think of is alanine. So beginning at A for alanine, and all it has is a methyl group there. That's the smallest hydrophobic thing you could stick onto it, right? So there's alanine, hydrophobic with just a methyl group. The next hydrophobic amino acid is valine, and if this were drawn in a more geometrically representative thing, that would be a V. So it has a V worth of hydrophobic stuff stuck out there. That's valine, Val, or V. You have phenylalanine, which is simply alanine, right? There's that methyl group with a phenyl ring. So phenylalanine makes sense from its name, and it's called Phe for phenylalanine, or F for the F sound. Here's leucine, and one way to think of leucine is it's a little longer than valine. So it's got an extra methyl group here, or methylene group here, and then the 3 carbons there. Here's isoleucine, also clearly hydrophobic, just a bunch of carbons and hydrogens, but it's isoleucine because of the rearrangement compared to leucine. So leucine is a symmetrical molecule, isoleucine is asymmetrical, and you can see how that's been rearranged. One way of thinking of it is that this methyl group has moved into there. Another hydrophobic amino acid is methionine. Now, thio, you should think that when you hear thio, think sulfur. So methionine is one that's got both sulfur in it and a methyl group. It also has a couple of carbons sort of laying the groundwork for that. So here's methionine. Pretty clear why it would be a hydrophobic amino acid. If you remember the hip casino mnemonic I gave you for electronegativity, sulfur is close to carbon in electronegativity. And so this is a fairly hydrophobic amino acid, but it has that sulfur in there. One of 2 amino acids with sulfur. Here's tryptophan, the largest of the side chains. Very clearly a big, bulky, hydrophobic side chain. It's no easy way to sort of fix that in your head except to learn that one. TRP for tryptophan makes sense. What's the W about? Well, we're going to see there are other things that T's been used for, so it's twiptophan. I didn't make up the code, but that was the mnemonic that was thought of at the time. Seriously. And then the last of the hydrophobic amino acids is quite a funny one. It's proline, and what's funny about proline is it has this 5-membered ring. So the side chain comes back around and makes a covalent bond to the amine, nitrogen. It still is capable of making another covalent bond somewhere, but it means that proline as an amino acid has a steric constraint on it that none of the other amino acids have. So we have this little small menagerie of hydrophobic amino acids that are similar but not identical. They're different in size. They're different in how aromatic they are. So for instance, tryptophan and phenylalanine are aromatic. The others aren't. This one has a heteroatom in it. So it gives some variety to build proteins out of. Now, in addition to the hydrophobic amino acids, we have some polar amino acids, ones that have enough charge difference that you have partial negative, partial positive charges. And some of them are quite obviously going to be polar. So here is serine, and serine is simply alanine plus an alcohol. And we know that alcohols are fairly polar groups, and so you could expect that alcohol group to participate in hydrogen bonding, for instance, or to interact well with water, for instance. Here's threonine, which is really just like serine, except it's got a methyl group added onto it. Okay. Here's tyrosine, and tyrosine looks like phenylalanine plus an alcohol group. And so it's aromatic, and the inner part of it, if you like, can behave as a hydrophobic side chain, but that alcohol group out there is going to behave in a polar way. One of the reasons for including it with the polar ones. We can have asparagine. So this is an amide group here. You've got, this is clearly fairly well suited to being a polar group, but it's just polar. It's not charged. Here's glutamine, which is like asparagine, but with another carbon in there. So without the amide group, it would just be alanine. Now we've got 2 carbons in the amide group. And then another odd heterocycle. Here we have histidine, or there are another heteroatoms. We have histidine. So this is a 5-membered ring with 2 nitrogens in it. And we're going to see that this histidine ring is actually capable of being ionized, although thinking of it as polar is a good way to keep it in mind. Here's cysteine. This is a methyl group here, so this would be alanine by itself with a sulfhydryl. Now it's a sulfhydryl, which is the sulfur analog of an alcohol, and hipocacino, the hydrogen is a bit separated from the sulfur, so it's a bit polar. It's not quite as polar as some other things, but it's certainly more polar than alanine. And here's little bitty glycine. Glycine does not have a side chain, it just has 2 hydrogens. So it's actually a symmetrical molecule. It's the smallest of the amino acids. It's not really clear that you could call it really either polar or nonpolar. It's so small its side chain isn't really even there. Any other questions? And then we have 4 charged amino acids, and I call these the charged amino acids because these side chains will be charged at physiological pH pretty much routinely. We're going to see some other side chains that can be ionized or not ionized. These guys are going to be routinely ionized at physiological pHs. So if you took alanine and added a carboxylic acid, you would have aspartate. You might remember we said we had asparagine. Asparagine has this amide. Aspartate has an acid. So there's a structural closeness between asparagine and aspartate. Glutamate has the same kind of relationship to glutamine. It's got 2 carbons instead of 1. So aspartate and glutamate are quite close to each other, but glutamate's just a little bit larger. Both of them are carboxylic acids, so they'll be negatively charged at physiological conditions in most cases. And then we have 2 that have side chains that are positively charged. We've got lysine. Now we've already used L for leucine, which was one of the hydrophobics. So the next letter after L is K, and there we have K for lysine. And it's simply this long chain of hydrocarbons with the amine group right there at the end, primary amine. In contrast, we have arginine. Now, we can't use A for arginine because we used A for alanine. So R for arginine is not too hard to remember. And it's got a somewhat shorter hydrocarbon chain here, just 3 instead of 4. And then this odd, really nitrogen-rich group called a guanidino group. So a lot of nitrogen in an arginine molecule. Aspartate, you can't call it A because we have alanine, so it's aspardate. Aspardate, as if you had a little bit of a lisp. And glutamate, we can't use G because we use G for glycine. So think of gluey. Things are gluey. That's the mnemonic. I didn't make them up. And that gives us 20 amino acids. There aren't really that many to learn. And they will definitely make life simpler for you if you have them at the tip of your tongue. I'm going to take a small break. I want to take a quick break to show you the JMOL versions of these, if I can. So those links that I showed you on the ICON homepages includes a link to the amino acids represented in the JMOL version. So you can look at them in space. And so you can have space filling, obviously a little bit less than easy to see what's going on there. You can do the ball and the stick. You can pick which amino acid you want to look at. Let's go to arginine, which is nice and big. Here's arginine. So there's a great big arginine. You can see that funny guanidino group. Very difficult to see on that one. Let's see, there's space filling. Guanidino group there at that end, the amino acid group there at that end. So I would suggest going through the amino acids here to get a little bit of a feeling for their shapes and so forth. Here's all of them represented as space filling right away. And then underneath it is a very nice table that shows you more properties as well. And we'll be coming back to that as we get there. Is there a question? Yes. Okay. Let me draw this on the board just for the moment. It's in you've got hydrogen and phosphorus are less electronegative than carbon and sulfur, which are less electronegative than nitrogen, which is less electronegative than oxygen. So this is going to show you the atoms from the least likely to hold on to its electrons to the most. Atoms that are over here will tend to pull electrons from atoms over here. And so a carbon-sulfur bond is going to not be very unbalanced. It will be fairly equal. Carbon-hydrogen is actually all right. Sulfur is enough farther away that this CH bond is a little bit polar. But obviously a hydrogen and an oxygen, there's a big span of electronegativity, and that's a fairly polar bond. Is that clear then? Does anybody like more? Does that make sense? Okay. Great. Okay. Okay. Back to amino acids. I want to introduce a concept to you that we're going to meet again in other molecules, which is to talk about chirality. And one of the interesting properties of most biological molecules is that they can exist in two or more forms in terms of mirror symmetries. And so when you get your gloves out, there's a left-handed glove and a right-handed glove, and you can't really put the right-handed glove on the left hand very well. If you simply were to describe them without referring to left or right-handedness, the descriptions would sound very similar, but in fact, they're mirror images. They're not identical. Mittens, on the other hand, go on either hand. This is a drawing to show you how an amino acid would be chiral. And this is showing you alanine, because you have a side chain here of a methyl group. And you can see that you can make that representation of alanine in either of two ways, with the nitrogen, amine nitrogen off to the right or off to the left. And so that's, in effect, a left or right-handed version of it. As long as you have four different things hanging off of that carbon, it can be chiral. It can be capable of having, will have, mirror image variations. Glycine, on the other hand, is not chiral, because with those two hydrogens, you can't tell one from the other, and so there's no way to have a left and right-handed. Glycine is the mitten of the amino acids, as opposed to the gloves of the rest of them. Now, some of these amino acids have side chains that do interesting things, and so we find them in certain places over and over again. Histidine has, I need to recharge this, let's see. Histidine has both an ionized and an unionized form. I drew it initially here. I can't remember whether I drew it initially as with or without the proton, but here's the unprotonated, unionized form, and it can become protonated or ionized. And those two forms both exist depending on the situation in the body. The pKa for this is pKa of 6. And so at pH 7, which would be neutrality, you'd have about 10% of the protonated form and 90% of the other kind. Now, what's interesting is that the pKa for the histidine as a side chain can be different in a protein than it is for the free amino acid. That a protein can create an environment that makes that histidine ring more or less likely to be protonated. And the fact that it can undergo reversible protonation at a range that's around kind of biological conditions means we will find histidine participating at the active sites of many enzymes because it's able to do that kind of reversible protonation. Cysteine is another amino acid whose side chain can be protonated or deprotonated. And so I showed it to you initially in this form, which is the protonated form and an unionized form. It can lose that proton and become deprotonated or ionized. Its pK is more basic, around 8.3. So it's generally going to be protonated at pH 7, but enough of it is deprotonated, again, to be useful in enzymes. An additional trick that cysteine can do is it can form bonds to other cysteines. This is called a disulfide bond. So instead of a bond to the hydrogen, here's a bond to another sulfur. And as you can imagine, when you have that kind of a disulfide bridge, you are going to tie together parts of a protein or tie together two different proteins. This is sort of a summary for you of some of the pKa's of these side chains. Both aspartic acid and glutamic acid have an acid side chain. The glutamic acid's pKa is a little more neutral. Of course, it has that somewhat longer side chain, so that affects how acidic that side chain is. We just talked about histidine and cysteine. Tyrosine, which was the phenylalanine plus an alcohol group, is also capable of losing its proton. Rather higher pKa than cysteine, so it doesn't do it very much, but again, it turns out to be handy in some enzymes. And then lysine and arginine are the ones that are positively charged in general under physiological conditions, especially arginine, because they're fairly good bases. This is showing you the diagrams of some of those structures. I think the ones in here that would be additions to what we've already talked about is reminding you that the ends of a protein have the potential for being charged as well. So one end of the protein is going to turn out to be a carboxylic acid. And as a carboxylic acid, it has a pK about 3.5. So unless something is done specifically at that end, it will, at physiological conditions or neutrality at least, be charged. And then there's an amino terminus to proteins as well, and it has a pK around 9, so it's going to usually be charged as well. But this is giving you the pictures of the charged forms. Okay. All we have so far are amino acids, which are building blocks to make something else. We haven't yet made any polymers. We need to put some of those amino acids together to make a polymer. And that turns out to be possible because the carboxylic acid of one amino acid and the amine group of another amino acid can be condensed against each other. The equivalent of taking out a water molecule. There's more chemistry to it than that, but you can think about it that way for now. So if you were to take out this oxygen and two hydrogens and create a new covalent bond between those two amino acids, you now have what's called a peptide bond. And this peptide bond is the basis for all the complexity of proteins. What's interesting about this peptide bond is that it is not a freely movable bond. That the carbonyl oxygen here and the nitrogen here, these atoms are interacting with each other enough that this bond has a partial planar character to it. It's sort of like it's locked in a particular plane. It's not free to twist around a whole bunch. There's enough interaction between all of those atoms that they create a unit. So you end up with a linear backbone of many amino acids joined by peptide bonds and side chains. Here they're represented as R1 and R2, side chains that then can stick off from this long peptide backbone. And this is just showing you several of these together in once. So you have 1, 2, 3, 4 residues here. These are the residues of the amino acids that went into it. You have a peptide bond here, peptide bond there, and there. Here's your amino terminus. There's your carboxy terminus. And you can see how that's something that could be extended a long way. There's at least one protein that has 1 million molecular weight as a single peptide chain. So it's a very versatile group. Now I can't remember if the J-mol, I don't think the J-mol amino acids shows you peptide bonds, but I do think that the first group I have there, the first link on the hemoglobin part includes J-mol peptide bonds. So this is a slightly better representation of why the peptide bond has some planar character to it. You can draw, if you're just drawing, the peptide bond at least 2 ways. So here you have both bonds to the oxygen and simply one bond to the nitrogen. If you were to rearrange the electrons in this, you could have it looking something like that. When you have multiple equivalent representations, it's often a clue that there's a measure of resonance there, and where resonance is behind, for instance, carboxylic acids and so on. And so here what this diagram has done is to try to sort of put each peptide bond on a little rectangular plane to show you that it's behaving as sort of a semi-rigid unit. However, this alpha carbon, so this is the central carbon of the original amino acid, the alpha carbon is not locked into things, so there can be rotation around this bond, and there can be rotation around that bond. So those 2 bonds off of the alpha carbon are what allow the peptide chain as a whole to rearrange and explore a bunch of different conformational space. Any questions? So here's another representation of an alpha carbon here, and it's then shown connected to two peptide bonds. There's one peptide bond there, another peptide bond there, and you can see that there's a finite amount of space that you can't have the atoms bumping into each other. And so there are some restraints on what kinds of shapes that proteins can take up, because just steric hindrance prevents a certain amount or certain domains from being explored. So far, we've only been talking about taking amino acids and putting them into a long chain to make a polypeptide. We've talked about it as a linear molecule. If we want to think about proteins in their mature state, when they're actually doing anything interesting, we have to think about 4 different kinds of structure. And what we've been talking about so far is often called primary structure. It's the simple sequence of amino acids. And so you can specify the sequence of a protein just by naming the amino acids one after another. That's a primary sequence. In many proteins, that linear sequence or primary sequence will take on another kind of structure, a local order where there's a local repeating pattern that's regular. And we call that secondary structure. We're going to look at 2 kinds of secondary structure, the alpha helix and the beta sheet. And we'll see how the hydrogen bonds within the molecule relate to that secondary structure. You can think analogously back to the nucleic acids. The nucleic acids have a primary sequence, a structure, where you just specify the sequence. But they can also have a secondary structure of that double helix. And you can even have tertiary structures. You might remember we showed you transfer RNAs that had a very complex, beautiful packing in space. Proteins also generally have tertiary structures where these little regions of secondary structure pack together overall to make an overall 3-dimensional packing of that polypeptide. the particular packing in space of that polypeptide. Now, some proteins go further, and they take more than one peptide and pack them together. And if that happens, you can have something called quaternary structure. So if you have two or more polypeptides that pack together into something, the way those tertiary structures pack in together is the quaternary structure of the whole assembly. So we can see here that we're going to see that myoglobin has tertiary structure because it has an overall packing in space, but hemoglobin is going to turn out to have quaternary structure because it's going to have more than one side chain in it. So. Okay, so let's look at a couple of examples of secondary structure. There are two that are especially important. The alpha helix and the beta sheet. And both alpha helix and beta sheet are going to be the result of hydrogen bonds that form between the carbonyl oxygen and the amide nitrogen. They're going to be capable of making hydrogen bonds within the overall polypeptide. The peptide bonds themselves are fairly planar, so they limit the possible rotation. And then of the ones that are permitted, the local order arises because of the hydrogen bonds that are forming between the carbonyl oxygen and the amide nitrogen. This is an example of the alpha helix. And let me just see one thing, how far we're going to go. All right. Yeah. This is an example of an alpha helix, and we're going to look at that in a little more detail as well. You can call that a ribbon diagram if you like. So the alpha helix is one of the two most common of the secondary structures. It is a right-handed helix, and I think the right-handedness of the helix might be just a little more obvious from that diagram. It's hard to know. There are about 3.5 residues per complete turn. It's about 5.5 angstroms from one to another. And the hydrogen bonds are running up and down along the long axis, along the axis of the helix. So the hydrogen bonds are pointing up and down to connect one coil of it to the one above it or the one below it. When you look at a space-filling diagram of it, it's quite clear that the center of that helix is in fact filled solid. This ball and stick diagram is a little easier to see the helical character maybe, but it doesn't sort of point out to you that the center of it's like a solid rod of atoms. And then the side chains stick out from the sides of that central rod in all the directions. So that's an alpha helix. The other most common of the secondary structures, the beta sheet, also depends on hydrogen bonding between carbonyl oxygens and amide hydrogens. But in this case, the backbone has been extended a bit. The backbone is pulled out more extendedly than in an alpha helix. And the hydrogen bonds are between adjoining or neighboring backbones. And so these are two of the ways in which beta-pleated sheets can be formed. This is called a parallel beta sheet because you have a carboxy terminus at each of the ends of this, or they're all running N terminus to C terminus parallel to each other. But you can see here the hydrogen bonds between the amide hydrogen and the carbonyl oxygen. Or you can have anti-parallel, where you have alternating directions between them, and in that case the hydrogen bonding is slightly differently arranged, but again can satisfy basically all of those potential hydrogen bonds and donors. Now you can imagine that if you're laying out the backbones like that, now what are you going to do with the side chains? Well, they're going to stick out either above or below the plane of the beta sheet. And I think we have a picture. Yeah, here's a picture for you. Here you can get a sense of how the backbone is at the center of all this, with the side chains sticking out above and below. And this is showing you that as a space-filling diagram as well. To connect these beta-pleated sheets, can either be from absolutely separate molecules, or within one molecule they can be connected either by a simple loop. If it's a simple loop, it'll tend to be an antiparallel. You can see the logic of that. Or in the cases of parallel ones, it may have an extended domain. It may be part of the molecule that's removed in the primary sequence from it by quite a bit. So we have primary sequence, the linear sequence, primary structure, the linear sequence of amino acids, secondary structure, the regular repeating patterns like alpha helix or beta sheet, and then you've got tertiary packing structure, the overall packing in space that accounts for all the atoms and within the molecule of protein. In general, in most proteins, most of the atoms, in fact, have locations. There are some known where they have floppy bits, but in most proteins, most of the atoms have a location. Early on, it was appreciated that proteins tended to behave either as if they were globular, compact packed things or extended fibrous things. And our two major tools for getting at that tertiary structure, that overall 3D packing, are either through NMR or through X-ray crystallography. Now, with X-ray crystallography, you need a pure protein, and this is actually here a picture of a crystal of protein, which is really quite strange when you think about it. When they first crystallized urease, which is the first protein to be crystallized, it was compelling evidence that a protein was a particular thing. It wasn't sort of an average of a bunch of stuff. It was a particular thing, because you can't crystallize things that are averages. And this is showing you how if you send a beam of electrons through a protein crystal, where the atoms are in that thing will scatter the electrons in very specific ways. And with enough ingenuity and math, you can take that pattern of little spots and turn it into a structure. This is showing you just a little bit of that kind of resolution going on, depending on the quality of the crystal, the quality of the X-ray beam, the detection device, and so on. You may have more or less resolution. And at the lower resolution, you have sort of an overall sense of where things are, but not a lot of specificity. Here you're starting to get the actual curve of the alpha helix, and by the time you get down to 1.5 angstroms, not only can you get the actual backbone through the alpha helix, you can get some of the side chains too. Now, there are some common features for proteins, and these rules are going to be fairly general. They'll get inverted when we get to membrane proteins, but for the moment, just think of these as general rules for protein structure. In general, proteins that aren't membrane proteins are going to have a hydrophobic center and a hydrophilic surface. So they're going to tend to pack, so the various hydrophobic amino acids like alanine and leucine and isoleucine are towards the interior. And then the surface will have hydrophilic side chains, which include both the polar and the charged ones, because that's going to make it easy for that protein to interact with water. Many proteins, although not all proteins, in addition to having the primary structure of peptide, have an additional piece of chemistry, another molecule in it, called a prosthetic group. And I don't know how many of you have heard of using prostheses and so forth, but a prosthetic group is simply a bit of chemistry that allows a protein to do something it can't do with just amino acids. The heme group is the prosthetic group for myoglobin and for the number of oxygen-binding proteins and for number of electron transfer proteins. The B vitamins are prosthetic groups for many enzymes and so on. And what we see here are 3 different ways to represent myoglobin. And they're each of them good at different things. This is the space-filling diagram, and this is, in a sense, the truest one in that this is the most direct representation of the surface that you might actually contact if you were another molecule. But it's kind of hard to see specific features in that. And so we've, over time, developed a number of ways of representing proteins, and these are a couple of the other ways to represent it. This is called a ribbon diagram, and in a ribbon diagram, alpha helices are replaced by these kind of spirals of ribbon because it's easier for our mind to notice that as a helix. There aren't any ribbons inside proteins, unfortunately. They'd be very pretty if there were. But it does help us to see the packing of those alpha helices because of their regularity. And so in the ribbon diagram, we can see the individual helices fairly clearly, and we can see that the heme group here is sort of tucked into a pocket. And then this is just a backbone diagram where it's just showing you the polypeptide backbone and lets you see the specific twisting and turning of that backbone in a more honest way, but a little bit harder to follow than the ribbon diagram over here. Now it can happen, especially as proteins get larger, that a tertiary structure can have more than one compact domain that has sort of an integrity to itself. So this is two examples of two-domain proteins. So you can see in this one that all of the green area is in contact with each other and fairly compact. And the red stuff is like that, but the two are only connected by this little slender strand between the two of them. Here the blue is compact and the red is compact, but just a little strand between them. In the case of multiple domain proteins, it's often the case that if you find a protease that can cut that strand there in the middle, that those domains will still have the function they would have in the intact protein. That makes it easier for studying. It's also a hint to us of part of how these things evolve, that you can have domains move between proteins to create new possibilities. I've mentioned that proteins tend to have hydrophobic amino acids towards the center. I've mentioned that we can divide the amino acids into hydrophobic and hydrophilic. It's not quite a black or white characteristic. And I don't expect you to memorize this scale, but I do want you to sort of take a look at it with me. This is trying to convey that things can be very hydrophilic or very hydrophobic. You don't have to think of it as just an on or off property. And if you look at the ones that are very hydrophobic, you can see here tryptophan, phenylalanine, and tyrosine, which is interesting because occasionally the tyrosine will have just that little alcohol group sticking out. And then at the hydrophilic end, you can see here that the various charged amino acids, here's the two carboxylic acids, and here's the two bases, and there's histidine. They're all quite hydrophilic. Very happy to interact with water. The differences in the in-between part are not nearly so strong. And this is, in this figure, what they've done is, I'm trying to see how well this works on your reprint. Let's see whether the color does it justice or not. Okay. So you may find this easier when you get back to your textbook. I can see that the Xerox is not quite so easy to look at. The red in this case, or the dark color on your handouts, is the hydrophilic residues. And the green there is going to be the hydrophobic ones. There are some residues like this one here, that aren't showing up as either hydrophilic or hydrophobic. So these are some of the amino acids, like glycine, for instance, that are not strongly favored for one position or the other. We can find them in both places. I see what time it is, and maybe we should pick up on protein folding on Wednesday. I'd be happy to answer any other questions people have.